So, welcome to lecture on protein structure basics for BK0001 Bioinformatics. Again, I'm Samuel Flores. I'm the course coordinator. This lecture should be a review for you, it's not strictly speaking bioinformatics, but I feel that a review of protein structure is useful for later understanding bioinformatical methods, especially methods that are related to protein structure. So today we're going to cover amino acids, and we'll talk about backbone dihedral angles, the structural hierarchy, primary, secondary, tertiary, quaternary structure. Talk a little bit about structure determination, and then about the protein data bank, or the PDB. Let's start with the protein backbone. So the backbone conventionally is drawn from left to right. So from N to C. So we start with the amide nitrogen, and the amide has its hydrogen, and then follows the alpha carbon, C alpha, and the alpha carbon has a side chain. Then follows the carboxyl carbon with its carboxyl oxygen. And then we go on to the next residue, which starts again with the amide nitrogen. It has hydrogen, and followed by its alpha carbon and its side chain, and finally its carboxyl carbon. So you need to know that the C alpha is a chiral center. That means that there are two energetically identical ways that it could place this a hydrogen on this alpha carbon. And to disambiguate this, you're going to typically assume that you have an L form amino acid. That's the predominant form of amino acids in nature. It's not that D form doesn't occur. It's just that it's quite much less common. So the way to disambiguate this is that uses Korn rule. So Korn means carboxyl, the side chain, and then the amide nitrogen. So if these three are placed clockwise like this, then that means that the hydrogen is coming towards you out of the page like this. You'll notice that in the following residue, you can read Korn going counterclockwise, and so that means that the hydrogen is going away from you into the page like this. You should also know about the dihedral angles. So there's two very important ones. Between the amide nitrogen and the alpha carbon, there's one called phi. And then between the alpha carbon and the carboxyl carbon, there's one called psi. Right? And these are going to be very important in just a minute. We're going to talk about how these are related to each other and how the side chains affect them and so on. So what are these dihedral angles I speak of? So a dihedral angle is a torsion angle about a bond. And it's defined like this. So let's say you're always going to define it based on these backbone angles, backbone atoms. The phi and psi are defined based on backbone angles, backbone atoms again. Sorry, and you're going to you can label them A, B, C, D. So let's say this carboxyl carbon is your A, nitrogen is your B, alpha carbon is your C, and then the carboxyl carbon is your D. So this is like A, B, C, and D. So your eyeball, or is like this. You know, the observer. You're looking down the bond between B and C, with B nearer to the observer, right? And then we're going to measure the angle that D makes with respect to A. So if you're the observer, you see this. And below that you see the B. And behind the B, can't see it, but it would be C. And then D is like this, right? So it could be at any angle. So here's this angle. This is the dihedral angle. In this case, we could say it's, I don't know, let's say it's 30 degrees or something. 30 degrees. So anyway, but the point is that it's measured clockwise from the A to the D, and it could be any angle, right? So if the D is hidden behind the A, then it would be 0, and it increases in this direction, right? And so we're going to come back to this issue of dihedral. It's going to be quite important. But I also want to tell you about groupings of amino acids, namely physical chemical classes. So this is a table of the 20 amino acids, you can put them into basically, roughly speaking, 4 main groups, right? The first group is the hydrophobic residues. You can see it's the largest group. So if you've got this, let's say this is your backbone. This is a carboxyl carbon. This is the alpha carbon, and this is the amide nitrogen. This is your side chain, right? So here you have alanine. That's the smallest of these hydrophobic amino acids. It only has 1 carbon in the side chain, right? And of course the 3 hydrogens. By convention, they don't draw the hydrogens. In fact, in this case, they're not even explicitly writing out the carbons. And then you have others, valine, isoleucine, and so on. There is, so the hydrophobic residues, some of them are very clearly hydrophobic, but there's some that are on the boundary. And one thing that you should understand is that there's really no such thing as a hydrophobic force. There's a sense, like water likes things that are not hydrophobic, that are hydrophilic, and we'll explain why. But it's not so much that that there's something pulling together the hydrophobic amino acids, as happens when you have hydrophobic collapse. It's more that that there's an opportunity cost to exposing a hydrophobic side chain to water, because it can't form hydrogen bonds, right? So water, if it's just pure water, it'll be making all sorts of hydrogen bonds, between one water and its neighbor, let's say, like that. But here, there's nothing I can do to interact with anything on the side chain of a hydrophobic amino acid. So it's basically that's a hydrogen bond that would probably be satisfied if it were exposed to solvent. So that's why it doesn't like being close to a hydrophobic residue. But anyway, the point is that that there's no such thing as completely hydrophobic, because nothing is completely nonpolar. We'll get into a little bit more about polarity in a second. But the point is that some people would not include tryptophan or tyrosine in the hydrophobic amino acids, because they're kind of close to the edge. So you'll see other tables where they actually, tryptophan might appear among the polar ones. So just know that there's a scale of hydrophobicity, and these guys, these last 2 guys on the right, are going to be more towards a polar scale, and some of these others will be much more in the hydrophobic scale, end of the scale. Okay, so then we move on to the polar side chains. And so anytime you see an OH, for instance, that's a clear sign you've got a polar amino acid, right? Because the oxygen is very electronegative, right? So it's got a partial negative charge. And then hydrogen is going to have a partial positive charge. So so that means that water also has this dipole. And so if there's a water molecule here, it's going to see that. It's going to say, oh, there's a negative charge. My hydrogen has a partial positive charge.
whereas my oxygen has partial negative charge. And so I'm going to point my hydrogen towards this oxygen, and then it'll be able to make this hydrogen bond, right? So it likes that. It can satisfy its hydrogen bond potential. Then another water molecule might come in here. I mean, it might even be the same water molecule. And it's going to look at this H. I'm going to say, oh, that's partially positive, and my oxygen is partially negative, right? And so it's going to point its oxygen towards that hydrogen, right? So there's two hydrogen bonds that it was able to make right there. Okay. Then we have charged side chains. So arginine and lysine, these are usually charged, right? Yeah, so always it's basically more or less permanently carrying a positive charge. Histidine is not always charged. It depends on the pH. So neutral pH is just neutral, but it could be charged. So it's classified in here, at least in this table, among positively charged side chains. Then we also have negatively charged side chains, the aspartate and glutamate. Lastly, we have these special cases. So they're special, each for its own reason. So glycine is special in that it's the smallest side chain, like basically no side chain, because you got the carboxyl carbon here, here's an alpha carbon, here's the amide nitrogen. There's nothing, there's no carbons, no heavy atoms attached to it. It does, of course, have two hydrogens, right? You could say the hydrogen is a side chain. So very small, and in addition to the very low volume, the consequence is that these phi-psi angles, which we defined earlier, these are very free. Like the hindrance that the side chain would have presented is gone, and so its Ramachandra plot, which we'll get into later, basically says that it's allowed to take on a much wider range of values compared to other amino acids. So it's basically going to have its own Ramachandra plot. So again, we'll get into this later, but basically most of the amino acids follow the same Ramachandra plot. Glycine gets its own, right? And actually proline also gets its own. So proline is special for kind of the opposite reason. Like, it's got a side chain, but it's special in that instead of being pointing out into the solvent or away from the main chain, it actually closes and is bound to the amide nitrogen. So that means that this bond, this psi angle, is locked, right? And it's actually locked in a position that is not compatible with secondary structure, which we'll get into later. But the point is that it can't make alpha helices, can't make beta sheets, so it's called a secondary structure breaker. Okay, and then the last special case is cysteine. So what's special about cysteine, it's got a sulfur here, as you can see. And so that means that it can make a disulfide bond. So it can lose its hydrogen and then bind to another sulfur in a different cysteine. And so now a chain that used to be maybe folded but only linked in a non-covalent way, well, now it's covalently linked, and so it's it'll contribute a lot of stability to your protein. It becomes very difficult to then unfold it. Okay, so that more or less covers it. So you should be able to given the name of amino acid, pretty much immediately know at least what physical chemical class it belongs to. Is it polar? Is it hydrophobic? Is it charged? You know, or is it one of these special cases? So the hierarchy of structure. So we have primary structure, and so that's what we have when we just have the sequence, right? And no structural information. So we know the amino acids. Let's see, alanine, leucine, isoleucine, lysine, arginine, whatever. I'm just making this up. That's the primary structure. Then you have sometimes you know that plus what sort of secondary structure it forms, right? So let's say, you know, the first, 10 residues make some sort of beta sheet, and then you have a few residues that are making an alpha helix, and then you have another beta sheet, right? So that's secondary structure. Tertiary structure is when you know how these pieces of secondary structure are positioned in relation to each other, right? So that would mean maybe you know that this is a beta sheet and that the alpha helix is kind of going across like this, and then there's the other beta sheet like that. Okay. This happens to be called the beta alpha beta motif, but whatever your structure is, the point is you know what it looks like in 3D. Then you might that's for one chain, and you might have additional chains in a protein complex. So let's say you have an identical protein, and maybe it's associating like this. So beta sheets like to form, well, like to associate like this, and so maybe this happened in this case, and so now you know how two monomers are related to each other, and that would be your quaternary structure. So here I'm showing you a homodimer. If there were different proteins, we would call it a heterodimer, and it could be, it doesn't have to be a dimer, it could be a trimer, quatrimer, any multimer. Okay, so now we've got the hierarchy of structure. And now we get into the Ramachandran plot. What Ramachandran did was he took something like this, you know, basically an argument that atoms can be represented by hard spheres, and asked himself what values of phi and psi are not allowed, basically. And so like I say, Ramachandran made his Ramachandran plot based on hard sphere arguments, like this model of like the electron cloud having this sort of van der Waals radius, and atoms not being able to invade each other's van der Waals radii, and that'll be, turns out it's going to be a kind of an okay approximation for these purposes. So okay, so let's start here with, this is the amide nitrogen, and this is the alpha carbon. This is the side chain. This is the carboxyl carbon. You can see the oxygen here double bonded to it. So the phi angle is the one around this, connecting these two atoms, right? Nitrogen to alpha carbon. And then the psi angle is the one between the alpha carbon and the carboxyl carbon. Okay, so the phi angle, it's always like your A, B, and C atoms that we mentioned earlier. Those are going to be always the backbone atoms, not the side chain atoms, right? So these ones that you see here, those are the ones that matter, right? So for phi, you would count the carboxyl carbon of the preceding atom, then the amide nitrogen, then the alpha carbon, and then the carboxyl carbon, and that's your A, B, C, D, right? So if we put this ourselves in phi equal to 0, that would require us to do something like this, right? Yes. So here you can see A, B, C, and D, and that's 0, right? So so if, remember that this peptide bond is fixed. I actually put tape so I don't accidentally move it. But so some things that are bad are like this angle here, side chain kind of interacting directly with the carboxyl carbon. And something that's really bad is if I do this, like that's basically impossible, right? So what angle would create that? Well, the phi angle here is something like 0, and the psi angle is something like, I'm going to go with maybe 180. See this? These nitrogens are on opposite sides of each other, so they're in trans. That would be 180. So 0 and 180. We'll go back to the Ramachandran plot in a second. But it's kind of useful to also look again at this side chain interacting with the carboxyl oxygen, and that happens at I'm going to go something like between 90 and 180. So let's say about 135 or so. Okay, so now that we switch back to the Raman-Chandran plot, we have some regions that are preferred, some that are prohibited, and the first one that we mentioned, that was we said that was the one where the oxygens are clashing, and that we said that was at phi is equal to 0. So it does matter a bit what psi does. So psi equal to about 180 is particularly bad, right? So that's this region. And then we go to this other one that we mentioned, and we said that phi equal to something like, what do we say, 135? Something here is not going to be so good, right? So some region around here. So that's just a couple of regions of the Raman-Chandran plot that are excluded based on kind of more simple-minded arguments. And then you have the allowed regions. So let's look at this beta sheet region. So we're going to talk more about both these pieces of secondary structures, but for the sake of argument, or for these purposes, you could say that the beta sheet looks kind of planar. Like it really isn't, but it's, like I say, for geometric discussion purposes, it's not a bad, terrible approximation to say they're planar. Where you have the backbone kind of doing this zigzag like this, you know, one here, up, down, and so on. And you can see that that's sterically that's pretty kosher. Like, you know, these carboxyl are pointing in completely opposite directions. This side chain is not near either of those. Everything should be okay. And you can already start to see that it's got some patterns, right? Like if you look at it like this, you see that you have an oxygen pointing one way, and then in the next residue is going to be pointing the other way. Actually, I should have said, let's say this is the preceding residue, and this is the next residue. They're going to be pointing in opposite directions. Same thing with the amide hydrogen. Like if you look at this hydrogen, it's pointing sort of this up here. Next time it'll be pointing down, and next time it'll be pointing up, and then pointing down. And I didn't show you any additional side chains, but perhaps you believe me when I say that if this side chain is pointing sort of towards you, the next one will be pointing the other way, and then towards you again, and then away from you. And so that's alternating. And it'll become a little bit clearer why I'm making a big deal out of that. Then we have another allowed region, which is this alpha helix region. And so the alpha helix is actually a little bit harder to generate. Let's say phi would be something like -90 would work. So maybe something like that. And then psi might be something like -45 or so. Let's see. So start here and then go this way. So this is one amino acid in alpha helix, and it might be hard to see right now, but it's kind of making a little C shape. The backbone is, and this thing, you know, you have to imagine that the next residue is going to be attached here, and so these things will make a spiral ultimately. These side chains are always going to be pointing out, and you notice that both of the carboxyl oxygens are pointing in the same direction, right? And both of the amide hydrogens, which I mean you're gonna have to imagine are attached here, both of those are pointing in the same direction. It's the opposite direction, right? That's going to be important. And then this thing has kind of a net angle. It's not pointing directly out. It's going to be pointing in a preferred direction. Okay, so we're going to, like I say, go into that in more detail. So here we are. This is the alpha helix, and so basically, if you have the amino acids in this configuration, you chain them one after the other. Like I say, they're going to end up having a net helicity, and they're going to have the property that all amine hydrogens. There's one. There's the next one. Or actually, I'm preceding one, I should say. Anyway, they're all pointing kind of up. Up in this case being the N-terminus. See, here's the amino terminus. They're all pointing up, away from the backbone, going away to the hydrogen. Okay, and then the oxygens, if you can see this, they're all kind of pointing down, right? And the geometry is just perfect so that this hydrogen can hydrogen bond with this oxygen, which is 4 residues before in sequence. And the preceding one will also attach hydrogen bond with one that's 4 residues before in sequence, and so on. And so every oxygen is at some residue N is hydrogen bonded to some amide hydrogen that's at residue N plus 4. And there are other secondary structures that are possible. It's possible to N plus 3 and N plus 5, but these are less common. I'm just going to do the most common one. And lastly, you notice that the side chain, it's pointing out, but it's a little bit pointing towards the amino terminus, right? This one's doing the same thing. So is this one. So is this one. So is this one. They're all kind of pointing up like that. And you have to imagine these side chains. They don't all look like that. They're not all alanines. Like they would most often have additional atoms. So maybe this is the beta carbon. This would be the gamma carbon, and it could have additional additional atoms hanging off. Whatever. The point is that these are longer, and so it ends up looking kind of like a Christmas tree. Like how a Christmas tree, it's a spruce has its branches kind of not horizontally straight out, but rather kind of sagging down. Another consequence of this kind of directionality that the helix has is that, going back to the issue of partial charges, oxygen again has a negative partial charge. The hydrogen is going to have a positive partial charge. So you have a dipole here. And all of these oxygen-hydrogen pairs also have a dipole, right? So here's a partial negative with a partial positive. And so if you have a whole bunch of small dipoles all aligned in the same direction, then that means you have a net dipole for the entire helix. And that has consequences for things like how it interacts with other secondary structure elements and how it gets inserted into the membrane, things like that. Okay. Now about beta sheets. So I told you that you can kind of think of it as being flat. For these purposes, so if we think about it that way, then we see patterns.
Here's the amide nitrogen, and then here's the alpha carbon. So they didn't mark it, but I think you believe me that this is the alpha carbon because there's a side chain. And then here's the carboxyl oxygen, and the pattern that you can notice pretty quickly is that if you look at the amide hydrogen, in the first residue is pointing up, and the second residue is pointing down, and the third residue is pointing up, and then down. So it's this up-down pattern follows, like it's always there in beta sheets along the length of the beta sheet. Then there's a carboxyl oxygen. Likewise, in the first residue is pointing up, in the second residue is pointing down, and the third residue is pointing up, and the pattern continues. So it goes up, down, up, down, alternating. Then there is the side chain. So here we're going to use our Korn rule to disambiguate which way it's going. So if you start with the CO and go towards R, that's clockwise. So that means that the hydrogen is coming towards us. So they didn't quite follow the best convention, I would say. But anyway, they tried to make the hydrogen darker to show that. But anyway, the hydrogen is coming towards you like that. Then in the next residue, the convention is the opposite, right? Korn goes like that, counterclockwise. So that means that the hydrogen is going away from you. And actually in chemistry, it's much more common to put a triangle like that with a small point towards the atom if the atom is farther away from you. So I'm going to draw it like that. So, but anyway, the point is that the side chain is now towards us. In the next one, the side chain is away from us. And then in the next one, it's towards us. Actually, they miswrote it here. It's not H, but rather R, the side chain. And then it's away from us. That would be like that. Okay? So the point is that the side chain is kind of pointing into the page in the first residue, out of the page, into the page, out of the page, into the page. So it's alternating along the length of the beta sheet. Right? And one way you can think of it is this. Like, you can, I told you that even though the beta sheet is not really flat, it's close enough to where, for the purposes of argument, you could say that it's a zigzag, right? And so, meaning that it changes direction. Like every bond, the next bond is going to be, like if after this bond this goes up, after the next one it's after the next one's going to go down, and after the next one's going to go up and then down. So you can think of this the dihedral angle as being close enough to 180 degrees to where you can say that's reversing direction at every bond. And since you have 3 bonds, then you have 180, 180. That would make a full cycle. But then you have another 180, so it ends up changing direction every residue. I don't know if you find that intuitive or if that helps, but anyway, that's how I think of it. And so, since we said that, let's start with this second residue. Its hydrogen and its carboxyl oxygen are pointing down. Then if you have another beta sheet and it's anti-parallel to the first one, this one is going the opposite direction. N is here, C is here. Then it's going to have exactly the same pattern. OH, O is an H going up and down, except it's going in the opposite direction, right? So first it will be the hydrogen and then the oxygen. Like the hydrogen will be on the right and the oxygen on the left, right? And so that means that and of course in both cases the alpha carbon is between them. So this is one one residue unit here, and this is one residue unit here. And so this is exactly this. These two residues are just basically rotate 180 degrees with respect to each other. And so this hydrogen finds it's perfectly aligned with this oxygen to be the hydrogen bond bond receiver. And here this oxygen finds the mean nitrogen of its partner residue to be perfectly aligned to be a hydrogen bond donor. Sorry, I should have said hydrogen bond receiver, acceptor in this case, not receiver, just to be a little bit more orthodox. Okay, and so we don't show any sheets up here, but then we do show one here, and you see it here again that we managed to satisfy the oxygens and the hydrogens. And so then if we move on to now officially move on to the second beta sheet, which is again anti-parallel. This is going to follow the same pattern as the one above. You have the side chain going into the page and then coming out of the page and then going into the page and so on. And as I mentioned, the oxygen is going up and then down, up and down. Same thing with the amide hydrogens. But what you notice is that let's say you were to continue this with even a third anti-parallel beta sheet. In other words, this third one is anti-parallel to the first. Then every single hydrogen bond donor and acceptor in this backbone would be would be engaging in a hydrogen bond, right? Look at this. So you got this hydrogen in a hydrogen bond, the carboxyl oxygen in the hydrogen bond, the next amide hydrogen in a hydrogen bond, and the next carboxyl oxygen in a hydrogen bond. So and again, these are very perfectly aligned, and so it's going to be able to make a very low energy hydrogen bond. It's just the ideal situation, and so anti-parallel beta sheets are very stable. Okay, and just as the direction of the side chains is alternating along the sheet, it's staying the same across sheets, right? So you notice that here you've got this side chain coming towards you, and then its partner here will also have a side chain coming towards you. Then here there's a side chain going away from you, and then its partner will also have a side chain going away from you. So the side chains are alternating along the direction of the sheet, and they're going the same direction across the sheets. And this is going to end up making a bioinformatical signature that you can detect, because the properties of these side chains that are close to each other have to be compatible. And we'll get into that when we talk about secondary structure prediction. So the third one, as I said, is anti-parallel to the second. So it goes from N here to C here. Again, it satisfies all the hydrogen bonds that are possible here between these two. And then the last one, the fourth beta sheet, that one is anti-parallel to the third, right? So if we start at start with nitrogen, here we're also starting with nitrogen, going towards oxygen on the right. And so this is following now exactly the same patterns as the one above it. So let's say that the side chain is above it is going into the page. This side chain is also going to the page. Let's see, this side chain is coming out of the page, then this side chain is coming out of the page. And now you can see that this carboxyl oxygen is actually aligned with the carboxyl oxygen above it. But what it really wants, of course, is to interact with a hydrogen, not with another oxygen. And so it looks for the nearest hydrogen, and look at that, it's a little bit off to the right. And the hydrogen has to look for an oxygen, and it's going to find it a little bit off to the left. So the fact that they're actually in the same, basically like exact copies of each other, but they don't want to look at each other. Hydrogen doesn't want to look at hydrogen, it wants to look at an oxygen, for instance. That means that they will not be able to make such stable hydrogen bonds, because this bond is longer and it's angled. It's not going to be as happy as it was in the anti-parallel case. This pattern will continue. So the conclusion is that parallel beta sheets are much less stable than anti-parallel beta sheets. And I wouldn't say that they're rare, but they're certainly quite a bit less common, less prevalent than the anti-parallel configuration. And my money would be on that. It's due to energetic reasons. Okay, and you'll notice that in the parallel case, just like in the anti-parallel case, you do have the pattern of side chains across strands going in the same direction. So I mentioned that already, going to the page and out of the page, and then into the page, and then out of the page, like that, and so on.
Okay, and then I want to talk about coiled coils. So I wouldn't exactly say this is secondary structure. It's more I would actually call it more tertiary structure. But in any case, a coiled coil is 2 or even 3 alpha helices that are in parallel and making contact, and they can start to wind around each other. So there's nothing that's kind of perfectly geometric about alpha helices. You remember it's not 4 turns, but rather 3.6, or sorry, not 4 residues, but rather 3.6 residues per turn. So there's always a little bit of this kind of like progressive effect in things like helices. And so as a result, if you have a regular pattern that enables these guys to contact each other, like in terms of integer multiples of steps, you're going to end up having some sort of spiral. And that's what's happening here. Like what you've got is some residues here that form a hydrophobic patch. And again, it's regularly spaced in terms of the number of residues. It's making contact with this hydrophobic patch here, and generating a net coiling for the two helices. And so if you want, so basically the way you can accomplish this is if you number your residues from 1 to 7, and you follow the following pattern. First, or let's say instead of 1 to 7 we say A, B, C, D, E, F, G, right? So residue A, the first one, would have to be hydrophobic. So that's yellow for hydrophobic, and then the next one would be polar, because that's going to be one that's pointing out into the solvent. So you want it to be hydrophilic. Then C, the following one, would be also polar, and then D would be hydrophobic, because now you're back into this buried hydrophobic patch. Then E would be charged, and the reason it's charged is because it's going to want to make some sort of strong interactions between these two residues that are not buried, that are going to kind of staple the two helices together, right? And so this one is going to have a charge, and it's going to be interacting with this one on this kind of partner helix. So blue was charged, and then I'm sorry, I might have said that wrong. Like after E was charged, and then F is the following one, and that one would have to be polar, and then finally G would also be charged, right? So we have these two charged ones, E and G, and on opposite helices, E would be looking at G, and G would be looking at E, and they would make a salt bridge, and these two residues, A and D, would be hydrophobic, and they would make a hydrophobic patch that would be buried, and then these remaining three residues would be pointing out into the solvent, so they would be polar. So so basically you have to have, basically if you want a coiled coil, you have to follow a heptad pattern. In other words, 7 residue pattern, and it looks like hydrophobic, polar, hydrophobic, charged, polar, and then charged again, right? And so you have 7 residues like that, and you have another 7 residues, and some number of repeats of this, right? And of course, you'd have to have a separate stretch of residues somewhere else that also follows heptad pattern. And then one would fall into helix, the other one would fall into helix, and the two helices would kind of wrap around each other in this coiled coil. So this pattern, it's pretty simple. So when you see coiled coils, you can even eyeball it and kind of figure it out. At least if you know that you're expecting to find a coiled coil somewhere in the sequence, you can actually, a human can actually see it reasonably. Not perfectly though, because it doesn't follow the pattern completely strictly, and also you have to consider things like it might have an insertion. Like sometimes it will have a little loop sticking out, and that will kind of that insertion inside the sequence will kind of shift the rest of shift the next heptad over, so you can't align it in an ungapped fashion. But like I say, you can look at some examples and see what that looks like. But a computer method can actually pick this up very with quite good accuracy and predict the coiled coils. Okay, so now the protein data bank, or PDB. So the protein data bank is by far the predominant database for macromolecular structures. There are others, but they're basically subsets of the protein data bank. So this is your go-to database for 3D structures. At the last count, it was at 210,000 structures, and of course growing. It's a little bit. I just have to say something about growth. Like people see curves like this, and just because everything seems to be exponential these days, they just automatically say, "Oh, it's an exponential growth." Not all growth is exponential. I just have to point that out because I've heard even PhDs saying this about this curve. This curve is definitely not exponential. Yeah. So some years ago, I wanted to actually figure out how fast this was growing, and so I could see that the rate of production was increasing more or less linearly. And so, as you know, the integral of a linear function is a quadratic. So I thought I'd fit a quadratic to it, and it actually fitted really well. So the growth, like I say, this was a couple of years ago. So it could be changing a little bit. It could be maybe there is a bit of a cubic term, but at the time the growth was very well modeled with a quadratic. So definitely a polynomial growth, not exponential. Nonetheless, there's a lot of structures in there. The reason AlphaFold, which we'll get into in the lecture, has been so successful is because it had so many structures to train on. So this is really good for machine learning methods. There are no new folds being discovered. The era of fold discovery is long past. So even though there are new proteins being found all the time, entirely new structural themes are not being found. Furthermore, there are some proteins that get more attention than others. For example, if it's a human protein, it's likely to get more attention than a protein from another organism. If it's a medically important protein, then it will get more attention still. And so there are some proteins that have been crystallized over and over and over again. You know, maybe just crystallized twice, maybe a lot more than that. And it's not exactly the same structure because maybe they managed to get higher resolution. Maybe it's in complex with some ligand. Maybe they mutated one of the residues. You know, there's always some reason they submitted. It's not. I wouldn't say it's redundant, but it's near redundant, and that actually turns out to be pretty useful for some methods. Like, for example, if you work on protein-protein interactions, as I've done, you find that you can do the same calculation on two different proteins, and you might say, "Oh, why are you repeating your calculation?" Well, no, I did it with different PDB structure, so it's different enough to where you can count it as a separate measurement. And as you know from the central limit theorem, if you add up your results from different measurements, your standard deviations sum in quadrature. So you end up getting a reduction in your uncertainty. In other words, your precision gets higher, your accuracy gets higher if you repeat this. So that's a nice thing to remember if you're doing any structural calculations, is you can always or very often repeat the calculation if you have more structures. How are these structures obtained? So the kind of the traditional method has been X-ray crystallography. We're not really getting into the method, but X-ray crystallography assumes that you have atoms in some sort of crystal, and then you have photons, X-ray photons, inciding on it, and then when they hit these atoms, the ripples come out from the atoms, kind of like around reeds in a pond, and then these ripples are going to add up either constructively or destructively at some detector screen, sometime far. And exactly how tall these peaks are and how far apart they are tells you something about how the atoms are distributed, and eventually you can determine how all the atoms are positioned in the structure. Then you have cryo-electron microscopy. So this has actually been a revolution starting some years ago in cryo-EM, and so basically the electron microscopes are quite old. The revolution came only from just better clustering. So what they did was that they started to realize that alluding to the point I made before about the standard deviation, the central limit theorem, if you have let's say you have a blurry shot of some protein. Let's say this is the ribosome. You can kind of see the outline of it. You can see some asymmetry. You can see maybe some very large scale gross features of it, but no details. Maybe you can find another picture of that same ribosome that looks the same way. That means it's sitting in the same way on that on your surface. So that means that you can just translate and rotate it and superimpose it, and then you find another one that's the same kind of, looks like the same kind of blobby shape, maybe in different rotation, but you can easily rotate and translate it and superimpose on that. And as you add up these images, the image starts to get crisper and crisper and crisper. Again, because of this phenomenon where your standard deviation of your observation tends to get smaller, like the square root of n, with n being the number of observations. And maybe this is one cluster, maybe there are several clusters, or many clusters, and those all represent different views of this ribosome. Like the ribosome could have landed on one side, it could have landed on the other side, it could have landed upside down, and all these views will help you determine the final 3D structure. And so that is, even though your initial view, single particle images are pretty blurry, it's amazing how much resolution you can get from this. Like there are lots of structures now with less than 3 angstroms resolution. Some of them even considerably better than that, and they seem to be getting better all the time. So that's good especially if you can't get a crystal. I mean, it has some limitations. You can't do small proteins. Like there has to be a minimum size to where you can see enough to where you can align the images. But the fact that you no longer need a crystal makes it really attractive for a lot of cases. And so it's especially good for very large things like the ribosome, or even bigger like viruses and so on. Okay, and then lastly, there's nuclear magnetic resonance, or NMR, and so this is kind of based on this this idea that if you have a magnetic field and you have some atom inside of it, the atom has its own spin, and so it precesses inside this magnetic field. It has, we know what its spin should, what its precession frequency should be. And then, this applied magnetic field is not the only field it feels. Like there might be another atom close by that might have the same or different dipole, and so that magnetic field influences the precession, the precession frequency of the atom, and so it shifts it a little bit. And so you can add up all these frequency shifts and control them until you're able to determine basically the distance between those two atoms. And so if you have all these distances between atoms, then you are eventually able to solve the structure. That tends to be better for very small structures. But it does have the advantage that you can do it in solution, so it doesn't, again, doesn't require crystals. The only thing is that there's so much experimental uncertainty with NMR that typically when you get an NMR structure from the PDB, it'll have several models in it. Like you'll see model 1, model 2, model 3, and you can play them in the movie. You can see kind of jiggling. Just don't make the mistake of thinking that's actually motion. It has nothing to do with motion. It has to do with the experimental uncertainty. Okay. And lastly, I want to talk about the format of these files. So the Protein Data Bank used to have a standard file format called PDB. So anytime you see a file with 4 letters .pdb, or 4 alphanumeric characters, I should say, .pdb, then it's in the PDB format. And that format, it was very convenient because it was so simple. Like you'd have different record types, and I guess the most important one was atom record, right? So the first 6 letters, it was fixed format. So the first 6 columns were for your atom record type. So if it were atom, it would be like the word atom and 2 blank spaces. And then it would have other columns, like let's say like atom number, maybe atom type, like CEO or whatever element it was. Atom name, actually maybe, no, sorry, atom type came later. This would be an atom name. Like we talked about alpha carbon, that would just be called CA. Talked about beta carbon, that would be called CB. and so on. And then you would have things like residue number and like residue name. Like so, it'd be ALA means alanine, LYS means lysine. So res name. I could be getting the order wrong, but you get the idea. Like there were certain columns that were reserved for each of these for each of these fields, and then there would be, most importantly, or very importantly, I should say, X, Y, and Z. So these are the X, Y, and Z locations of that atom, and there would be occupancy and some others. But the limitation of this is something that was not apparent early on. I mean, early on, when the PDB started, we're looking at proteins that maybe look like myoglobin or something like that, where they're all a certain size. Maybe a few hundred residues max, and so this format suited that just fine. But what happens if you have a number of atoms that's a very large number of atoms, where these characters that you set aside for the atom number are not enough, because you max out at 99999 number of atoms, whatever, and then you add one more, and there's no fields left to express that number, right? So that's it. You reach the upper limit of your ability to express this protein in this format, right? Like maybe you could make a different PDB file or something like that, and people did that, but that seemed kind of awkward. Also, same thing happened with residue numbers. Like if you have more, like a very, very long chain, maybe you run out of residue numbers. One thing that happened a lot actually is that there's just one column for the chain ID. And that was fine back when people were working with monomers and dimers and trimers, but along comes something like maybe DNA origami, where you have a gigantic structure made of DNA, and one of them is very long, like this backbone kind of scaffold, sorry, but the rest of them are very short, so-called staple strands, and they're just hundreds, maybe thousands of them there could be, and so if you have only one column for the chain ID, then that means that you have your alphabet letters, and you have your 10 digits and maybe some punctuation, and then you just run out of letters like that. Like you cannot no longer express name additional chains. So that was a problem. And then in terms of your coordinates, X, Y, and Z, well, you had a fixed number of columns for each of these coordinates. And so what happens if your structure is very large? Again, like DNA origami, let's say. So that means that the bigger you get, you have to cannibalize your precision, right? So maybe initially you had several places for precision, so you could specify thousandth of an angstrom if you wanted to, but then once your structure gets big and you need more integers for the integer part, you might get to a point where there's only maybe a tenth of an angstrom precision left. Like you can only have one digit after the decimal point. Maybe you could have 0 digits after a decimal point. And at some point you might not even have enough room for angstroms, and then you're done. So there was a problem with this being fixed format, limited your precision and the size of your numbers. So along comes MMSIF, and actually it came along quite a long time ago. But it basically solves all of these problems. What it does is it defines some sort of there's one more problem that I didn't mention, which is that once you have fixed column format, what happens if there's an additional property of this atom that you want to express here? Just who knows what it could be, but something that doesn't fit into these fields, then you'd be out of luck. And people would make up like extra new extensions that were only supported by one professor. Actually, I did that actually also. Like I came up with ways to hack this limitation on the number of digits in your coordinates. But it's hacky. Like you can't have that as a sustainable system, right? So extensibility was a problem. No extensibility, basically. So anyway, so along comes MMSIF, and it addresses all these problems. It also comes with a new problem, but it does fix all the ones that I named. Firstly, it's no longer fixed format. Like now you still have columns, or rather fields separate fields for the different quantities, right? So you could have, let's say this x, this is y, and this is z. You know, and they're separated by space, but they're no longer fixed, right? If I wanted to, I can make this x longer. I can put in as many significant figures as I wish. And I can do that with x, y, and z, and I can make x have lots of digits and y have only a few digits if I want to. Like, you know, the point is that it's no longer fixed format. And so that means also, again, I can have unlimited atom numbers. My chain ID, which would be, actually, I'm not sure if which one is the chain ID. One of these two is the chain ID, but I could have. I'm not limited to just a single letter. I can name my chain ID like, you know, Donald Trump II or whatever. You know, I can make it arbitrarily long as there's no spaces in it, right? By the way, not a supporter of Donald Trump, in case that wasn't clear. Let's see. So yeah, and so basically, there's no more limit on precision and size of numbers and length of identifiers and that sort of thing. And then going on to the extensibility issue. So what they did was that they had like, if you have a block of atoms, you would start by defining what fields you're going to provide, right? So here you're saying, okay, first I'm going to give it this, you know, just a field name atom, and then atom site ID. That means like atom number. That's these guys here, and then followed by the atom type. So PO, you know, basically periodic table, what element that is. And then a longer identifier that actually names this atom. So this is actually a nucleic acid, so it's OP1. It's one of the oxygen atoms on the phosphorus, and then OP2 is a second oxygen atom on the phosphorus. Then there's O5 prime, C5, all those other guys. Then another important one is X, Y, and Z. Occupancy 1. So like this is occupancy, just like if you're uncertain, there's two locations where the atom could occur, like, you know, because thermodynamics allows that sometimes. Then this might be not 1, but, you know, half of it would be here and half of it would be there. Like, okay, and so basically now it's extensible, because if I wanted to, I could create an additional field. Like, let's say for some crazy reason I don't want to look up my periodic table, so I just want, like, you know, mass number, right? Or whatever. I could add the mass number as an additional field. The problem, and it's really kind of a trivial problem, or it sounds like a trivial problem, but it's enough to where I think it's caused a significant delay in adoption of MMSIF, is that it's just a more complicated file format, right? Because you have to think about, you know, like make sure this matches that, right? So you could get two different MMSIFs and you try to combine them, like to make, let's say you want to add a molecule from here to this molecule. Before, like with the PDB format, like you had a bunch of atoms, you could just copy paste, like cut them out of the second file and just plop them in at the end of the first file, and then you could open up in a viewer and you see both molecules work perfectly, right? But here, if you did that, like, you know, you just have to make sure that this block matches, right? Because if you have a different number of fields, then it gets completely messed up, right? So it's not, I wouldn't say it's hard to do, but it's just one additional step that has to be done. And as a result, you might be surprised at how recalcitrant people have been to abandon the PDB format. There's still software being written that only supports the PDB format, even though it's been declared dead. My software does now support MMSIFs, but I have to confess it took a while. Okay, and I think that will be it for our basic protein structure lecture. And so, as you know, you have a reading that's assigned. So you do that, and then you will have a quiz, which is to be turned in prior to our discussion. Once we get into the discussion section, then you should be ready with all your questions, and we'll answer them at that time. So these are just a few pictures of people in the bioinformatics section. And with that, I'm going to sign off.